Okay, with that analysis done, now we have our data back. So of the enhancers that we looked at that have CPG data available that was measured by this assay, we have a little under 1900 regions that are significantly methylated, and we have a little under 600 that are unmethylated. So this is about a 23% unmethylation rate in enhancers, and I'm interested in those. So as is the case when you have about 600 data points, the first thing you want to know is, well, what genes are these near, and is there any sort of significant enrichment? Are there any patterns in the data that we can glean? So one of the tools that was developed at Stanford to answer this exact question is GREAT. And GREAT is a tool developed in the Bejarano lab to look for potential functionality in different genomic regions relative to background. So we'll get to this in just a second, but the first thing that we want to do is to go back into our data and take it from Galaxy and now to save this back to our computer locally. So I'm interested in unmethylated enhancers in this case, and so I'm going to click on this now anachronistic hard drive disk in order to download this locally, and I'm going to do the same thing for all of the genomic regions that are my enhancers as a background, because I'm interested in, relative to a background of enhancers, is there anything specifically interesting about unmethylated enhancers in this cell line. So I'll save these as well. And now when we go to GREAT, it will take us and ask us, do we want, which genome assembly do we want to use? Where are the test regions? And so in this case, the test regions that we're interested in are unmethylated enhancers, and the format that these files are in are just bed format, so chromosome start stop, and that's actually very important that they stay in that configuration. The background region, feel free to use the whole genome. It's much easier to do this way because there's specific rules around what your background can be, which is that your background must be a strict superset of whatever your test regions are. So that in this case, every region that's in my test region is also in the background. You can't have any sort of distinction there. So if GREAT throws an error, that's oftentimes what it is. But we'll use all of the K562 quote enhancers as our background. And the only other thing now that we need to change is the association rule. So we're associating a genomic interval with a gene, and we need to know how to make that mapping. This is a matter of preference, and feel free to use the default, but personally I like to use the two nearest genes, and I use a 120 kb window as outlined by some of Job Decker's work, that many of the enhancers fall with putative, I should say, regulatory regions that touch promoters are about 120 kb away. So from there I'm going to submit. And GREAT is going to be processing our data and pulling up enrichments in a number of different categories for potentially interesting biology with our data. So it's going to be running here and loading our data in. And once that stops, it's finished. And then you have a number of different categories which are represented here. So we're talking about gene ontology terms, and then a number of different metrics by which you could find potential enrichment. I'm going to say that it's very important that you do not need to find something that's necessarily statistically enriched. In this case, I'm going to be going into the pathway commons because this was found to be our number one enriched pathway. In this case, it's NFAT signaling. However, I want to show you that if you have no terms at all in any categories, which can be common when using GREAT, you can go into Global Controls, and you can just click Ignore Statistical Significance, and this will give you, for example, the top go terms. And even though they're not statistically enriched, this will give you something to potentially go off of. So the other thing that I'll mention that is nice about GREAT, which is that it will give you a region gene association. So if you use this for nothing else, you can get an export of all of your regions and what that nearest gene is and how far away it is, or the nearest two related genes. But from here, I want to know about this enrichment for calcineurin-dependent NFAT signaling. So you can click here, and it will take you into some interesting data, which is that here are all of your regions. In this case, they're unnamed, but if you included a fourth column, in addition to chromosome start-stop, that had some sort of denoting name for your different regions, they would be listed here, and then what genes they're associated with. So here we have a gene-based mapping, and if you click on the gene itself, what it will do is it will pull up a UCSC genome browser track. And if you had been working previously on the same computer on the UCSC genome browser, it will remember all that data, and it will load in custom data for you. So in this case, these are the background regions denoted here in red that we uploaded as K562 enhancers. Here is the test region that we found to be enriched that is not methylated. And if we zoom in, what you can see here on the methylation data, where a more orange color denotes methylation and more blue is unmethylated, is that you have a number of regions here that are potentially methylated. The transcription start sites are unmethylated, and then this enhancer is as well. So that is the end of this tutorial. Have fun sifting through your own data, and we'll be looking forward to seeing you soon.